happy that all these technical problems have been solved. And I'm also very happy to be here tonight and to talk with you about VIRIDIC and VirClust and how to put things into taxonomic boxes, how to put viruses into taxonomic boxes using these two tools. Basically, they work more or less on the same principle. Both of them, they calculate some type of intergenomic distance. For example, VIRIDIC is based on nucleic acid, VirClust is based on protein clusters. And using this distance, the viral genomes are ordered hierarchically into trees, and later on, different user-defined thresholds can be used to split these trees into viral clusters. Before going into details, I would like to give a brief overview on the different hierarchical clustering tools or methods that they are there. So we can split them in two. We can split them in those which are based on multiple alignments or on those which are based on pairwise alignments. Basically, anytime you have genes or proteins which are similar to each other and can be multiple aligned, you can calculate pairwise distances between the genomes based on these multiple alignments and calculate trees based on these distances, trees which are able to capture evolutionary pathways and distances. And this is great. Unfortunately, there are no universal genes for viruses, as we all know, or whenever we are talking about single genes, the trees might be biased because of horizontal gene transfer. Whenever we have genomes which are more distantly related to each other, and we want to put them into a tree, we are doing so-called pairwise alignments, which compare the genomes 2 by 2 and calculate distances based on this alignment. And a result from this type of alignments is that sometimes a distance in between organism 1 and 2, for example, can be based on a completely different region as the distance between organism 1 and 3. Using this type of distances, again, we can calculate hierarchical trees, but these trees, this time, although they are capturing the similarity between viruses, they don't necessarily capture evolutionary paths and distances. And to start with VIRIDIC, VIRIDIC is using a simple method to calculate intergenomic distances. It's basically taking the number of identical bases between two genomes and normalizing them to the total genome length. And this was Andrew Kroplinski's favorite method for calculating intergenomic identities between viruses for a long time with the purpose of classifying viruses at the species and genus level. And at some point we met, we decided to work together, and this is how VIRIDIC was created. In comparison with other similar values, as for example, average nucleotide identity, VIRIDIC again normalizes the identical bases to the genome length, but ANI, average nucleotide identity, normalizes to the alignment length. That means that whenever the two genomes share only short regions of homology in between them, the ANI can grossly overestimate the identity between the two genomes. Once VIRIDIC calculates the intergenomic identities, it can output them as clustered heat maps, and you can see them here in the upper panel. And in the lower panel, we have different indicators of the alignment quality, and I can talk more about this in the questions and answer session. Using the distances created by VIRIDIC and the trees used to cluster the viruses, we can split the viral genomes into species and genus level clusters, for example. And here, default values that VIRIDIC is using are 95% identity for species threshold and 70% identity for genus threshold. I want to point out that these are flexible thresholds and they can be user-defined to better accommodate the different type of viruses that need to be classified. And to shortly compare the VIRIDIC identity value with the thresholds used in Viromics so far. So looking at the Viromics paper, we can see that 95% ANI over at least 80% of the genome alignment has been usually used to delineate species level viral populations. If I do the mathematics, this would basically correspond to 76% intergenomic identity as calculated by VIRIDIC. Right, so to move now to the second tool, to VirClust. This tool has a much more complicated workflow compared with VIRIDIC, but it can be shortly summarized to take input genomes, make protein clusters, and then cluster the genomes based on their protein content and calculate the core proteins between the different genomes, and finally do protein annotations. To start with the protein clustering, in the beginning, VirClust takes the proteins from all viruses and compares them using BLASTP and puts them into clusters based on the BLASTP similarity. These clusters can then be compared based on their hidden Markov model profiles and put together into protein superclusters. And if we do a third level of clustering by comparing the HMM profiles of the protein superclusters, we can then obtain protein superclusters. Right, so it seems that this protein clustering is quite important and it will actually impact all the outcome of the analysis, of the VirClust analysis. And because there are different parameters which can influence the protein clustering, I have made them all available and adjustable from the interface. So different protein clusters can be created depending on the many purposes of the study. For example, clusters can be created from very stringent, where only identical proteins are found in the clusters, to very relaxed clusters where the proteins are similar at the HMM level. Right, so we have the distances, we have the protein cluster content of the different viral genomes, and we can use this protein content to calculate distances in between the different genomes. And the way to calculate that, it's rather simple, because it's a distance, we say 1 minus the shared proteins in between two genomes divided to the total proteins that the two genomes have. Once this distance is calculated, then it can be used to create again hierarchical trees. And again, these trees can be split into different clusters depending on the different distance thresholds used. These clusters will represent different taxonomic levels, or can represent different taxonomic levels. And VirClust has quite a lot of outputs, so many tables, many protein files that it gives back to the user, either to be used directly by the user or to be inputted in different bioinformatic pipelines. My favorite output from VirClust is this integrated visualization, where I can see side by side the virus tree with the heat map showing me the protein content of each individual viral genome, where I can see some genome-specific statistics. And also I can see the different viral genome clusters as created based on the different thresholds I have used. I also like this visualization because as opposed to just looking at a simple tree, Now I can see the proteins, so I can see the features which are responsible for the clustering. And if, for example, in this viral genome cluster, one of the viruses would have a completely different protein cluster content, I would immediately know that this was a misclassification. So I would investigate further, or I would test other methods to produce the trees. Once I know the viral genome cluster, so once I define the viral genome clusters, I can use VirClust to calculate the so-called core proteins, so the proteins which are shared between all the genomes in one viral cluster. And I can extract these proteins and I can use them further to calculate phylogenetic trees. At some point, I was curious how well the VirClust hierarchical clustering compares with the ICTV classification. So I took a subset of the ICTV viruses, mainly Caudovirales, not Podomyo and Cipoviridae, but all the other families and few other non-tailed virus families. And I put them in VirClust and I performed hierarchical clustering. In this case, illustrated here, both based on protein clusters and protein superclusters. What I can notice is that the hierarchical clustering produced by VirClust is matching quite well the ICTV classification. And in the quest of finding a threshold which could be used to cut the VirClust tree into clusters of, for example, family level, I compiled this table and I saw, for example, that the distance threshold of 0.9 works quite well to define families in the Caudovirales order. However, the same distance can only be used for defining viruses in the Adenoviria realm, which is something to say that, yeah, no threshold to rule them all, as we have heard from Evelyn before. And because I actually have developed these tools mainly for my students and for my associates so that they can actually characterize and classify their many virus isolates, I want to give here a short example of one such case study where my associate, Nina, has isolated more than 100 viruses, and we have used VirClust to order them hierarchically and to group them into 10 new phage families. And here I would like to point out that in this study, we have also compared the VirClust clustering with the VIP3 and Victor-based protein trees, and they were quite comparable. So, how about the availability of these two programs? They are both available in the internet as web services with graphical interface at viridicicbm.de or virclust.icbm.de. Just to point out that VirClust is still work in progress, so our servers can misbehave quite often when we are running VirClust, and I would like to ask you to be patient and to give us feedback. Both tools also available as standalone singularity distributions, and the one for VIRIDIC is out there, and as I have heard from the user community, is working quite well, and VirClust, we are working to prepare it and it will be available soonish. And to think about help with these two tools, first of all, Andrew Kropinski, which was extremely enthusiastic about the development of VIRIDIC, and he was very supportive, and also did the complete benchmarking of the VIRIDIC paper. Also, Arvind, who helped with part of the benchmarking analysis. I would like to thank the members of my lab, the students, and associates, which are heavy users of VIRIDIC and VirClust, and criticizing and bringing problems quite often in front of me so I can solve them. And not in the least, I would like to give a big thank you to the user community, which was quite helpful with the feedback, and it helped me polish both VIRIDIC and VirClust. And I think I'm well in time, or I hope I'm well in time. And right now I'm ready for questions. Thank you.